Okay, so this is still chapter 14. So again, we're talking about DNA structure and function. And this is part 3 where our focus will be on DNA replication. So how it replicates will be our focus. Okay, so first off, when does DNA replication happen? So we learned way back in chapters 10 and 11 about mitosis and meiosis. And we said before either of them can happen in interphase, in S phase of interphase, all of the genetic material, the DNA, has to be copied. And so that's when DNA replication happens, before a cell is going to divide by either mitosis or meiosis. Okay, so that's the when. Now the question is how. And so, again, giving you the historical perspective, if we take a look at the DNA structure, so the model of DNA, it makes sense that the two strands would separate from each other. So the bases, those hydrogen bonds between the bases would break, and the two strands would separate from each other. And that both of those strands would then serve as a template to make a new complementary strand. That makes sense, just looking at the structure of the DNA. And in fact, Watson and Crick and Rosalind and Wilkins, that's what they kind of thought would be. But they didn't know that at the time. And so there was actually 3 hypotheses that were generated and then tested. And so the 3 hypotheses were DNA can replicate conservatively or semi-conservatively or in a dispersive fashion. And so let me show you what these 3 models looked like. Okay, so again, these are the 3 suggested, proposed models for DNA replication. And so the first model all the way on the left, here is our double strand of DNA represented by these 2 kind of gray bars. And it was suggested that these come apart, 2 new strands are made, those come apart, and then these 2 strands, the original, again shown in gray, come back together, and the 2 new strands, shown here in blue, are together. So that's conservative DNA replication. So the original double-stranded DNA molecule is conserved, and the new DNA that's generated through replication would be together over here. So we'd have one double-stranded DNA here and a second double-stranded DNA here, 2 copies. That's a possibility. That's called the conservative model. The second model that was proposed is called semi-conservative. So again, in gray, we have our original 2 strands of DNA here. They come apart, and each serves as a template. And in the end, we end up with one double-stranded molecule here and a second here. And if we look at both of these, they're made up of 1 strand of old or original DNA, shown in gray, and the other strand, shown in blue, is the new DNA. So 1 old, 1 new, 1 new, 1 old. So again, this is called the semi-conservative model. Lastly, the dispersive model, again, here's our original DNA shown in gray, and the dispersive model says that there's a mix of old and new DNA in both strands as the result. Okay, so it's old and new DNA is dispersed within the 2 strands of the molecule. And so this was tested. So here is the test. And so the scientists that did these tests were Meselson and Stahl. Okay, and so this is famously known as the Meselson and Stahl experiment. Okay, so here's what they did. So first off, the model organism that they were using was E. coli. Okay, wonderful model organism for this experiment. E. coli is really easy to grow in the laboratory, right? You can grow it in broth culture. You can grow it in a petri dish on agar medium. Very easy to grow, and it replicates very rapidly. So about every 20 minutes, new E. coli are being formed. So you can the experiments that they did, they needed replication to happen pretty rapidly, and you can get that with E. coli. Okay. So they used E. coli as their model organism, their model system, and this is the other important part. They used 2 different isotopes of nitrogen. And so remember, DNA is made up of nucleotides, and one of the components of a nucleotide is a nitrogenous base, right? Adenine, thymine, cytosine, and guanine. And so when we say nitrogenous bases, they contain nitrogen. And so we could get E. coli to take up these isotopes, these different isotopes of nitrogen, and we can use that to our advantage. And so that's what Meselson and Stahl did. They grew E. coli in the presence of these isotopes of nitrogen. That nitrogen will get incorporated into DNA, and then they can do some analysis because of that. So the two isotopes of nitrogen that they used were N15 and N14. And so N15 has, one more neutron than N14, and so it weighs more, right? It has a greater atomic mass. And so because of that, N15 we're going to call heavy. It's the heavy isotope, where N14 is going to be a lighter isotope. So we're going to say that it's light. And if we were to try to separate out DNA in a density column, DNA that has heavy nitrogen is going to go to the bottom of the density gradient because it's heavier. The N14 containing DNA is going to be lighter, and so it's going to go towards the top of our density gradient. So they use those differences in density of N14 and N15 to their advantage. Okay, so again, they have E. coli, and they're going to really label the DNA with different isotopes of nitrogen. Okay, so let's look at the actual experiment. So again, why nitrogen? Why did they use isotopes of nitrogen? Because DNA has a lot of nitrogen in it. So it's a great way to really tag that DNA. They started the experiments by growing E. coli for several generations in culture, in medium, containing heavy nitrogen. So they had, an Erlenmeyer flask, which is pictured here. They had broth culture. They had E. coli in there. And the only source of nitrogen that the E. coli had was this isotope, N15. And so if we looked at the DNA of this E. coli, it would all be heavy DNA. Okay. They then took a little bit of bacteria from the N15 flask and put it in a new flask containing just N14. And they let the bacteria grow in this flask over a few generations, isolating DNA after each generation. And they could time it perfectly because they know E. coli double in about 20 minutes. So it was a very controlled experiment. So again, they harvested some E. coli after each generation of growth. And they took this out a few generations. And so let's look at, hold on a second, the next slide. Here's some more of an explanation, then we'll look at the actual, some pictures of the experiment. So with each generation, they would isolate some of the bacterial cells and they would pull out their DNA. They would then take that DNA from those separate generations and put it onto something called a density gradient. Okay, and let me actually draw this. So I got my pen here. I'm going to draw a test tube to the best of my ability with this mouse. Why that happened. So here's my test tube, and inside of it, change my color. Oh, I don't know, purple. Inside of it, I put a density gradient. Okay, and a density gradient usually is made of something called cesium chloride. Not a big deal. It's a density gradient, so we've got kind of a liquid in here, and now I'm going to add DNA. So if I added DNA from my bacteria that had just been growing in my N15 culture, all of my DNA would go to the bottom, and I would get a nice thick band down here. Ooh, I don't know why that does that. So all of my DNA here, if it's at the bottom, it's all heavy, N15. Okay. Now, if I wait just a generation, I would expect this to shift up. Okay. So I don't know why that does that. Sorry. This band here would represent DNA that's made up of both N14 and N15, heavy and light, some of both, okay? And then up here, if I have DNA that goes way, way up here in my density column or my density gradient, this is going to be DNA that's made up of 2 strands that only have N14. So that's very light DNA. Oop. Should be a 14. I accidentally drew a 15. A touchscreen computer. Okay, so heavy DNA would go down here to the bottom. Light DNA would come up here and settle at the top. And DNA that's made of a mixture of heavy and light would end up here in the middle. Okay, and if we go, up in increments, this would be lighter and lighter. Okay, so we kind of have a basic understanding of how a density gradient works. It's going to separate DNA on the basis of density. So heavier DNA is going to go to the bottom, lighter DNA is going to go to the very top, and DNA that has kind of a mixture of light and heavy is going to end up kind of in the middle. So let's look at their experiment. So again, they started off by growing E. coli in this flask right here that just contained heavy nitrogen, so N15. And so all of the DNA of this original population or original generation would have all heavy DNA. So if we come down here, this is at generation, 0 or time 0 before we do the transfer into the N14. All of the DNA, if we put it in the density gradient, all of the DNA from this original culture that has only seen N15, all the DNA is going to have just nitrogen 15. And so we're going to get one single band thick down here towards the bottom of the density gradient. So all of the DNA is heavy. We then take some DNA out of this culture and put it into this culture. So this culture over here on the right side contains just the light nitrogen, so N14. And so we allow the E. coli to replicate just one generation. And so we're on to the first generation here. So time 20 minutes. And what we see when we pull the DNA out and we put it on our density column is we see a shift. So all the DNA that was just right here, this nice thick band, what we find is this shift. Now all of the DNA is a little bit higher in the column. 100% of it, here's a nice thick band. So what does that tell us? That tells us that now the DNA, all of it, is made up of a mixture of heavy and light nitrogen. And so right off the bat from that first generation, we know that the conservative model is not true. Okay, why? So if the conservative model were true, then we wouldn't have this migration of the band to the middle of the column. Instead, we would have one band up here made of just light DNA, and we would have one band down here made of just heavy DNA. So this would have been the original, and this would have been the new. And so one of the models we can already throw out. Now the other two models, the semi-conservative and the dispersive model, are both supported by the first generation. Where's my pointer? Okay, so generation 1, having the band in the middle tells us that the DNA is made up of both heavy and light. Again, both of those models show heavy and light. So what Meselson and Stahl did is they took it out to a second generation. And this second generation and beyond are all evidence for what we call the semi-conservative model. And so in the second generation, they let E. coli grow a little longer, so a second generation out, in just the N14 media. And what they found is they get 2 bands, one in the middle. This would represent DNA made up of one strand of heavy and one strand of light. And then they get one up here, which is 2 strands both made of light DNA. Okay, and as time progresses, the band in the middle gets slimmer, which means, a lower amount of DNA. And then the band towards the top gets thicker. So it makes sense. Let me show you this another way. Okay. Same exact experiment, just drawn kind of differently, and sometimes students understand this picture a little bit better. And so again, we've got three models that were proposed: the conservative model, the semi-conservative model, and the dispersive model. And actually, before I go through this, let me just back up here. Just want to remind you of what those models look like. So the conservative model, again, we start off with two original or old strands of DNA. These would both be made up of N15 if we're talking about the experiment. And after a round of replication, we would end up with one double-stranded DNA molecule that was still made of just N15 and one double-stranded DNA molecule that was made up of just N14. So off the bat, if this model were correct, after the first generation, we would see a split. And we would have 50% of the DNA down at the bottom of the density gradient, made up of just heavy DNA, and 50% of the DNA up at a higher band, made up of N14, just light DNA. We don't see that in their experiment. And so after the first round of replication, the conservative model gets thrown out the window. It's not supported by the evidence. Okay. The second two we have to take out to the second generation. So the semi-conservative model, we would start off with two old or original or heavy strands. Okay. After one round of replication, the two double-stranded daughter molecules, the result, would be made up of one heavy and one light. And so the data supports that, right? So we start off with all of the DNA at the bottom. We let replication happen once and boom, the band moved up. And so all of the DNA is in the middle now of the column. And again, that represents DNA that's made of a mixture of heavy and light. Well, we see the same thing here in the dispersive model. Both daughter strands are made up of both heavy and light DNA. They're just arranged differently. So again, it's the second generation that shows us that the dispersive model is not true. If the dispersive model were true, then the DNA in the middle, after that first generation, would just slightly move up with each round of replication. But that doesn't happen. We end up getting two distinct bands, one in the middle and one at the top. And the top band gets thicker, with each round of replication. So again, let me take you back to this slide. And again, sometimes it takes students a little bit of time to understand this. This is a very important model to understand. You may see this on an exam. Okay, I like this question. So here we go. On the left down here at the bottom shows our three models and the predicted results. Okay, after the first round of replication and after the second round of replication. So one generation, two generations. And so we'll start up here at the top at number 1. So this Messelson and Stahl, the scientists, took E. coli and grew them first in a culture containing just heavy nitrogen 15. So this is a flask containing bacteria, again, grown in just N15. So all of the DNA, both strands, is going to be heavy. So if I were to put that in a density gradient right now, it would go to the very bottom. Okay. They then took some bacteria out of here and transferred it to a new flask that had just N14 in it, so light nitrogen. So now the DNA is going to start to incorporate light nitrogen. They took some DNA out after about 20 minutes, which is just one generation, and then they let some stay in there. Okay. They put that DNA into a density gradient, and what they found is that all of the DNA from this first generation went to the middle of the column. And so that's going to be evidence that the DNA is made up of one light and one heavy strand, or one original and one new strand. Okay, so let's come down here to predictions. If the conservative model were correct, then after the first round of replication, you would have two bands, one at the top showing the new DNA made of just light nitrogen, and one down here towards the bottom showing the old or original DNA made up of two strands, both containing heavy nitrogen. That is not what they found. So off the bat, we can cross that out. It's not true. However, again, the semi-conservative and the dispersive model In both cases, you can actually see they're drawn, right? So the old or original N15 strand is drawn in dark blue, and the new N14 strand is represented with light blue. And so both daughter strands, or all the DNA from this first generation, would be made up of one heavy and one light chain, and therefore you end up with one band in the middle. The same is true for the dispersive model. You would end up with just one band. So both of these models are supported by this evidence from the first generation. So luckily, Meselson and Stahl kept going. They left some E. coli in here, let it grow another generation, pulled it out, and this is what they find. They find out that there's still a band in the middle, so still a band made up of DNA containing one heavy and one light strand, but then there's a second band up here at the top containing DNA that's just made up of light nitrogen. Okay, and this is where the dispersive model also gets kicked out. So let's come down here to the dispersive model first. If this were true, then what would have happened after that second generation is this original band would have just shifted up a little bit because more N14 would be incorporated. But we don't get that. We get two distinct bands, one still in the middle and one at the top. Well, that is what the semi-conservative model predicts. Okay, so the band in the middle is still going to have DNA that has original and light, and the band at the top is going to be made up of just the light. Okay, and with each round of replication, again, this band here at the top is going to keep getting heavier, I shouldn't say heavier, larger and larger, and this band here in the middle is going to have less and less DNA. Okay, so again, the model that is correct is called the semi-conservative model of replication. And again, the scientists who did that work were named Meselson and Stahl. It's their famous experiment where they used different isotopes of nitrogen. And so here I just wanted to end this part of the chapter or presentation showing you, again, this model. So this is what it says. It says you have a double strand of parental DNA. And what happens is that parental DNA separates. Okay? And so the hydrogen bonds between those bases in the middle of the molecule come apart. They break. When the DNA is separated, these two parental strands serve as templates, which means that they are read. And the order of the bases is going to dictate how the new strands are made. And so again, here in light blue, I'm showing you the new strands, which would have been N14 strands. Okay, and if we look at base pairing rules, in the original parental DNA here, you have an adenine, and in the new DNA, you're going to have a thymine, A to T, C to G. Okay, and it happens in a semi-conservative fashion. Okay, that is the end of this part of the chapter.